Good afternoon, everybody. My name is Jason, and today I'll be presenting to you my research on exploring the role of norepinephrine in epilepsy. So, for starters, I'm going to begin by going through my background / literature review, followed by my research question, my methods of experimentation, my findings through experimentation, and I'll wrap everything up with the conclusion and any questions that you may have. So, for starters, what is epilepsy? Epilepsy is recurrent brain activity which comes in a synchronic pattern. As you can see on the far left, we have a normal single action potential that is firing from a brain. Now the difference between a normal brain and that going through an epileptic seizure is an epileptic seizure has the same action potential, but it's occurring in a pattern, and it's a much more frequent pattern. So I mentioned to you what an action potential is. Now, an action potential is a stimulus that triggers a neuron to first begin to depolarize the cell. What I mean by depolarize is the sodium channels within the cell open, which allows the sodium, which is a positive ion, to flood into the cell. This positive influx of electronegativity into the cell causes an action potential to occur at the very peak, and prior to the peak the sodium channel will close, which will then open up a potassium channel. Potassium is another positive ion which is inside the cell. After the action potential is reached, potassium will flow out of the cell, which will cause the cell to go through a hyperpolarization, which means the cell is becoming highly negative because its positive ions are flooding out of the cell. After a certain point, the potassium channels will close, which will then cause the cell to go back into its resting potential, waiting for its next action potential to occur. And again, an action potential is a brain signal that's going from one neuron to the next neuron. So for the research that I've been working on, we've been primarily focused on the hippocampus. Now the hippocampus plays a large role in attention, memory, and learning. And within this hippocampus, we have different subregions. There's the CA1, the CA2, and the CA3, and much more. For our research, we've been focused on the CA3 region of the hippocampus. So, going back, we use, within the brain, there's different types of neurotransmitters. There's dopamine, norepinephrine, there's serotonin, GABA, and much other neurotransmitters. What a neurotransmitter is a chemical that binds to a receptor within the brain. And for us, we're looking at norepinephrine as the neurotransmitter that we'd like to really investigate. And we want to know what norepinephrine is involved in to mitigate seizures, since we found that norepinephrine plays a large role in regulating seizures. So norepinephrine plays a large role in attention and memory. And it also has anti-epileptic properties, which I already mentioned, and we're looking at norepinephrine's role in the CA3 region of the hippocampus. So within the CA3 region, there's different adrenergic receptors. There's the alpha and the betas, and within them, there's different subcategories of those receptors. So there's the alpha 1, alpha 2, there's the beta 1, beta 2. And for our research, we've been looking at the alpha 1A adrenergic receptor. Primarily because prior literature has showed that alpha 1A looks quite promising for for regulating seizures in a controlled manner, to which would leave a human being actually able to function throughout the day. So questions that we were primarily trying to answer is which receptor is responsible for norepinephrine's anti-epileptic properties? And also, which of these receptors would be quite promising to target, so that we can formulate a drug that will target these receptors and create another pharmaceutical that can be used to better treat epilepsy. So in order to do this, we use electrophysiology as our method. And what electrophysiology does is it measures and records brain activity. And we're able to use electrophysiology to record brain activity in select neurons. And to see which receptor was primarily responsible for the effects that we were getting, we had 3 different types of mice. We had the wild type mouse that it's a normal mouse, has complete normal brain function. Then we have the alpha 1B knockout, which does not have the alpha 1B adrenergic receptor. and then we use alpha-1A adrenergic receptor knockouts, which again does not have the alpha-1A. And since we're primarily interested in the alpha-1A, we wanted to see what activating that receptor would do in a mouse that doesn't have the receptor versus a mouse that has the receptor versus a mouse that has the receptor but it also doesn't have the alpha-1B. So we're just using that to primarily see what our results were getting us. And also, in order to mimic a seizure, there's numerous different types of methods, but for ours we decided to take out the magnesium that was in cerebrospinal fluid. And if you take out the magnesium, magnesium plays a role in regulating seizures. So if you get rid of magnesium, neurons will actually go through, it'll induce a natural seizure in a neuron if magnesium is not present to regulate it. And we also used, to target the alpha-1A adrenergic receptor, we use phenylephrine, which is a derivative of norepinephrine, and I'll get into in a minute why it's similar and why we were able to do that. So on the far left, we had phenylephrine, and on the right, we had norepinephrine. Now they look quite similar, and the only difference between the two is that there is no hydroxyl group on the phenylephrine, which norepinephrine has on the phenylrine, and also phenylephrine happens to have a methyl group attached to its nitrogen, whereas norepinephrine only has a hydrogen in the place for the methyl group. And by using phenylephrine, we're able to still target the alpha-1A adrenergic receptor and use that as an agonist to mimic norepinephrine. Now, phenylephrine does not cross the blood-brain barrier, so phenylephrine cannot be used by itself as a pharmaceutical to target in the hippocampus. So what that means is phenylephrine, when it goes into the blood system, it cannot cross from the blood into the brain. Now, phenylephrine can be used for the heart, which is used in medications for the heart, but it cannot be used for neurological disorders such as epilepsy. So in order to do this, we had to use neurons that were placed on a electrophysiology stage. So rather than being within the animal, we were doing in vitro studies. So we were actually we were administering the drug directly to the neurons rather than through administration, like oral administration to the animal. So what we got is the beta adrenergic receptor. When it was activated, we happened to get an increase in epileptiform activity, so seizure-like activity, which is not what we want. So as you see here, we have our control prior to getting the activation of the beta receptor. And as time went on and we increased the concentration, we got more and more frequent seizures, which is horrible for anti-epileptic medications. So now with the alpha-2 adrenergic receptor activated, we see that this is what we begin with, normal epileptic activity. And then we have a significant decrease in activity to where we're only having a single action potential occurring, which you may think this is awesome for epilepsy. We've stopped the seizures. But this actually mimics the drug atipamezole, which is used in veterinary surgery as anesthesia. So if a human being was on this type of medication, they would be asleep. They couldn't function. So we had to look elsewhere, which is where we come to the alpha-1A receptor. So with the alpha-1A adrenergic receptor, we have a baseline of the seizures occurring, and then as we administer the drug, we see we have a slight decrease in the gap of activity. So right here we have the gap, it increases in length and increases in frequency. So this actually looked quite promising to formulate a drug to be able to control seizures. We would hope that with this type of drug, we'd be able to treat epilepsy while also leaving the person able to function throughout their normal day. And that's the current problem with anti-epileptic medications. They reduce memory, they reduce learning, they leave you comatose and tired and unable to go through your normal day, which to function in normal society, we want to be able to be normal. So with the alpha-1A, it looks quite promising to be able to continue the study into future testing to see if this drug would actually be more beneficial than current drugs on the market. And as I mentioned earlier, anti-epileptic medications now reduce learning, they inhibit memory, whereas this drug, we found that in prior research, we had a mouse that we called super mouse, and this mouse had the alpha-1A adrenergic receptor always activated, And this mouse had increase in memory, increase in learning, and decrease in anxiety, which hence why we call it super mouse. This mouse was amazing. So if this drug is anything like what it does to super mouse, this is in a way a super pill that we'll be able to make. So, in conclusion, we found that activating the alpha-1A led in a decrease in epileptic activity, whereas if we have the alpha-1A knocked out, we have no change in epileptic activity. So by activating the receptor, we have decrease in seizure activity, and not activating it, where the seizure is going on. So for future research, we'd like to continue our experimentation with the transgenic mice. However, now we're going to be getting new mice from Johns Hopkins. These mice will be a little different than what we've been using, because the knockout mice that we had been using, their receptors, the alpha-1A knockouts, did not have the alpha-1A ever. So from the time of its development onwards, it never had that receptor. So anything that came with the receptor during the development, so say a different receptor was supposed to come, but it didn't because that receptor wasn't intact to be able to allow for the additional receptors to be formed. These new mice will be normal mice up until the time we give them a chemical compound that will deteriorate the alpha-1A receptor. So everything that comes with the alpha-1A during development, those mice will have, and we'll be able to selectively deteriorate that receptor. So it'll make our results a lot more clear, and we'll be able to understand it a lot better than how we do now. And next, we'd like to also be able to synthesize a drug that can actually cross the blood-brain barrier so that we can take our testing into in vivo. So rather than using phenylephrine, we'd like to use a drug that we can administer to the animal and see it actually take effect and see what this drug will actually do. And last, like I said, we'd like to carry on these testings into in vivo. References. I'd like to acknowledge my mentor, Dr. Van Does, for helping me along with this project. The grad student, Joe Bigany, this is actually his PhD project that I've been working alongside with him on. He's taught me everything I know about neuroscience, because as a chemical engineer, I don't know anything about neuroscience. So I'd like to thank Patrice and the rest of McNair for helping and making this possible. Any questions?
Why did you choose to study this? Was it just the guy you were working with, or did you have an interest in it? Or?
Going through the interviews with McNair, they asked me, well, what are you interested in? I said, I want to make drugs. I like pharmacology. So sent me over to Dr. Does, because he does a little bit of pharmacology, and this just happened to be the project that he's working on. So so I got a little bit of what I wanted to do and learned a lot more of extra stuff that comes along with pharmacology. So
I can't remember, how much biology did you have before?
I've taken enough for pre-med. So I have up to cell biology, which cell biology is why I was able to understand all this.
Okay, that's what I was wondering.
Without that class, I wouldn't know what an action potential was or how neurons communicate or anything about them. So, and then the organic chemistry helps with the actual drug itself. Okay.
This is kind of off in another direction, but could you potentially see the part where you talked about attention being used for hyperactivity in children? Like I know a grant is funded for epilepsy, and so you wouldn't go there under a certain grant, but I could see a path.
So talking to Dr. Does, because I'm interested more in psychological disorders, so that was one thing I'm interested in, and this drug actually gives a decrease in anxiety. And I know that a lot of epileptic medication is used to treat anxiety and depression. So for this to be used for ADHD, I'm sure that this receptor, we may be investigating it primarily for seizures, but it could be used for so much more. Yeah, so any neurological disorder, this could be associated with it. So right now, it may be an epilepsy, but where it goes in the future, that would be interesting. So.
And then also, like, where you, in the beginning, where you got the results that you did not want to get, is there any way to suppress, go the other direction and use that research that went exactly opposite, to
Yeah,
Is
so,
that in a positive way to, I would say diluted, or,
so, yeah, lots of this can be used for so much more. So like the beta, like that, the type of data that we didn't want to get, that was already, we were aware of that through literature, and then through experimentation we were able to see that. But even though we may not want that, that data can still be used for a different disorder. So, I forgot what it's called, but to where you're always exhausted and tired, I forgot what that disorder is called.
fatigue?
Yeah, it's like the chronic fatigue.
Narcolepsy?
Narcolepsy. Oh, sorry. So that would be, possibly this could be used as a stimulant. If you were to target the beta adrenergic receptor, you can stimulate the body to be able to function a lot better, which may not be good for epilepsy, but there's so many other disorders that this can be used for. So just by understanding the receptor and everything that comes with the receptor when you activate it, we could pick and choose what we want to activate and for which disorder it'll be used for. So.
You said one of the mechanisms that you used to completely get rid of the epilepsy with the people, or the, not people, but the mice who were completely like asleep.
Mhm.
Do you think there's anything you can maybe add to that, like, I know this probably would not work, but like caffeine or something to keep the person awake but at the same time get rid of that?
So to stimulate it, yes, you can target different receptors, and the drugs that are currently on the market, they don't necessarily target one receptor. They target a whole range of receptors, and then once you target one receptor, everything that comes with that being activated, it's a chain reaction. So like you're saying, to use caffeine. Now the beta, the alpha-2 may not be the best bet for that, just because it's so powerful. But if, say, the alpha-1A gave a sedative effect and we didn't really want that, we can accompany that by targeting another drug that may be producing adrenaline. So rather than a downer, it's an upper that can complement the effects of the other receptor.
Yeah, well, that's kind of what I'm asking.
Yeah, so that definitely could be a possibility. Unfortunately, we're so far from possibly getting this on the market to be able to test it in humans or animals. So the first step now is to get a drug that'll cross, and then we can really see what's going to happen if what we see on in vitro testing will actually follow through in vivo.
My sister had a brain tumor, and I watched her go through seizures toward the end, and the quality of life goes down pretty drastically at that point. Do you foresee, any positive benefits, for terminal brain tumor patients?
I don't know right offhand. It's
Because a lack of seizure is what, the goal would be.
yeah, because the seizures they like to say that they don't cause damage over time, but the severity of them they do once you've had so many. So if you could reduce it, I would say it would really help. Maybe for a terminal patient who's really suffering from this, maybe a decrease in the seizures or to control seizures may reduce the progression of the disease. It may not prevent it, but it may suppress it for a much longer period of time. I would hope so, though. Anybody else?